Okay, so welcome everybody. Thank you for the introduction. So I continue very much along the lines of David's talk, and I will talk about the intra-tumor phylogeny problem that has already been introduced, and sort of I give you my perspective on it, let's say. So the way we got into this, and also in a way probably my main motivation for this is more in a clinical setting. So I'm not an evolutionary biologist, and we work for several years with people both in Zurich and in Basel on the molecular tumor board, where we have set up computational pipelines to analyze molecular profiling data, DNA and RNA from tumors, in order to bring these information eventually into the tumor board and into the decision making for the treatment of the patient. And the basic setup is that, of course, that you take a biopsy, you sequence DNA, RNA, whatever, and then there is a number of steps that involve a lot of computational and statistical problems, identifying genetic alterations in the tumors, so mutation calling and so on, and then interpreting these changes. What is a driver? What is a passenger? What does it mean for the phenotype? Is it predictive of any clinical outcomes, and so on? And these results are eventually compiled into a report that really a computational biologist brings into the tumor board, and together with other experts they decide on the treatment. So for the students here, you may end up sitting in a tumor board at some point in your career, so you better be prepared and you know what you're doing. Good. So and there's a lot of statistical modeling here. So one of the major problems in this process, there's many, but a major one is this huge diversity and heterogeneity that exists on a molecular level between tumors from different individuals, but also within tumors. And really that's, for me, the starting point to look at this in more detail and to try to understand and to model this diversity. Before I start doing so, there's one or two reminders why this is very likely to be very useful. Also, I've been asked this in the lunch or coffee break this morning, well, why are we building these trees anyways, right? What's the point? And yes, there is enough basic research interest from an evolutionary biology point of view that in a way would probably already be enough. But I think it's already now very clear that they will be useful for diagnostic, prognostic, and eventually probably also treatment decisions. So this is a very early paper addressing this, actually not based on next generation sequencing, but still on Sanger sequencing, where we just look at the incidence of a certain carcinoma of the esophagus. And there's two groups. One group of tumors that show a high genetic diversities, which is quantified here simply by the number of clones that have been identified, and another group with a low diversity. And you see that depending on the diversity in the tumor, you have an increased chance to progress to the disease. So this is independent of what the mutations are. Just the fact that you have more diversity within the tumor is a predictive marker for poorer outcome. And maybe more surprising, also the order in which mutations occur can matter for the outcome. So this is similarly the progression of a certain myeloproliferative neoplasm. And there's mutations in these two genes, TAT2 and JAK2, and depending on the order in which these mutations have occurred in individual patients, you again have a vastly increased chance of progressing to the disease. So the diversity matters, and also the order in which mutations occur matters a lot. So not being evolutionary biologists, we look at the evolution of cancer in a more naive way. We just think of it as genetic changes accumulating in the population. And the tumor phylogeny problem then looks like this. So you start with your heterogeneous tumor. So it consists of cells that are distinct genetically, indicated by the different colors here. And they are defined by different mutational patterns, indicated by these little dots inside the cells that are supposed to visualize the mutations. So the evolutionary history may look like this. So you have to read the timing from top to bottom. There's the mutation initiating the tumor on the very top. It gave rise to this growing clone. New clones come up, some of them go extinct, and eventually other mutations give rise to these other clones. And then at the bottom of this diagram is sort of the time point when you take a biopsy, when you sample the population. And we are only interested for now in the single cell situation. So I'm taking a finite sample in the form of a finite number of cells that I'm obtaining at some point from the tumor. So the data looks like this. It's, let's say, almost a binary matrix where the rows are indexed by the cells. The rows are indexed by the mutations and the columns are indexed by the cells. And you put a 1 if a certain mutation, a certain DNA base change has occurred at that position for that particular mutation in that particular cell, and otherwise you put a 0. Of course, there can be missing data as indicated here already, and the red colors already indicate that there can be false negatives and false positives, so this matrix in general will not be perfect. There are errors due to amplification and sequencing. So the goal now is to reconstruct the phylogenetic tree that I will call the cell lineage tree. So that's a root to tree for us, where the tips are labeled with the individual cells, and the interior vertices correspond to common ancestors of these cells, in such a way that if you trace out a path from the root on the top to any of the leaves, you're picking up a certain number of mutations. The mutations occur on the edges of the tree, and the cell that you're reaching is genetically defined exactly by the mutations that you pick up on this unique path. So that's the very basic setup we will make throughout the infinite sites assumption. But rather than with the cell lineage tree, for reasons that will become clear in a moment, we usually prefer to work with the mutation tree. So mutation tree means that the vertices are now mutations rather than cells. But other than that, it's interpreted in the same way. So if you trace out a path, you accumulate mutations. And in fact, the cells can be attached to the mutation tree. And in that manner, you would eventually also reach a cell. And it again is defined genetically in this manner. So under the infinite sites assumption, these two representations are equivalent. You can go back and forth. And in a way, it doesn't really matter. But for computational purposes, it does make a difference whether you operate on the object of the left or on the right, depending on the number of cells and the number of mutations that you have in your problem. In particular, and I will make it precise in a moment, the mutation tree is much more efficient for large numbers of cells and moderate numbers of mutations. If it's the other way around, you're better off with the cell lineage tree, but the main application we have in mind is a large number of cells, and then you can only do a moderate number of mutations. So that's really what we are targeting, and then the mutation tree is the better object to go with. Another thing that I want to emphasize is that most of the time we will not care about the attachment of the cells, and that is mainly because usually for these single cell analyses you have no additional information about the cells. You destroy the cell when you try to get the DNA and sequence it, and at least for the standard protocols you cannot do anything else with these cells. So you have no additional information, and that means you're not really interested in the relationship between individual cells. So it's not like a classical phylogeny problem, human, mouse, dog, and you want to know, is our closest relative, is it human, is it mice, or is it dogs, or is it chimpanzee? Because you know something about these species, that question makes sense. But here the question, does this is this cell closer related to that one or to the other one? That question doesn't make any sense. The question that you try to answer here is, well, what is the general structure of this evolutionary process? What is the pattern? And what is the order and the branching pattern in which mutations have occurred? And for that, it doesn't matter where the cells are exactly attached here. Okay, so the model that we call site is, in a way, I would argue, the simplest model you can use for the tumor phylogeny problem. And I will explain in a moment why. So let me first show you the model. So suppose you have M cells and N mutations. By T, we denote the tree topology. And by sigma, the vector of attachment points for each cell. So for each cell you have to tell to which mutation in the mutation tree you're attaching it. So then there's only two continuous parameters, the false positive and false negative rate, alpha and beta. And then you can already write down the likelihood. That's the probability of this binary matrix, maybe with missing data. And it just factors over all cells, and it factors also over all mutations. And then you just have the probability of the entry in your matrix, Dij, given the expected value. And the expected value is given by the tree topology and the attachment of the cell. And it's just given by this confounding matrix where the false positives and false negatives enter. So it's the simplest possible model in the sense that you only have the branching order of the mutations. There's no notion of a branch length. There's no explicit DNA substitution model, anything like that. It's much simpler. But it does account for errors, for observational errors in the data. Because they are so prominent, it's something that you cannot ignore. If you ignore that and you just use a standard method, it will be very misleading. Also, you might think, well, a simpler model would be to have only one parameter rather than alpha and beta. But as we will see in a moment, alpha and beta are typically very different from each other. And again, it would probably be not a good idea to use only one parameter here. So I said already, we don't care about the attachment points. So here are two mutation trees, identical mutation trees with different attachment vectors for the individual cells. So the way to deal with this probabilistically is to marginalize out the attachment. And that means you have a big sum here over sigma. So that means over all vectors of attachment points for all cells. That looks like a very bad thing to compute because that sum grows exponentially and there would be no way to do this only for a moderate sized problem. But luckily it factors again over cells. So really you have to do this attachment individually only for each single cell. So then this computation becomes feasible and just becomes linear in the number of cells and also linear in the number of mutations. So this is sort of a standard trick when you compute these marginal probabilities along a tree, Felsenstein algorithm, whatever you want to call it. It's the same type of trick. It factorizes along the structure of the tree and allows you now to compute the marginal likelihood independent of the attachment of the cells in time that's linear in the number of cells. So for inference, we are left now with finding the optimal tree. That's a discrete object. So the search space is this set of all trees. And if you have n mutations, the size of that space is n plus 1 to the power of n minus 1. That's the real problem, to find the optimal tree in this space. But the good news here is that the size of this space does not depend on m. It does not depend on the number of cells anymore. It only depends on the number of mutations. So for each likelihood computation, we need linear time in the number of cells, but then the search space is independent of the number of cells. And in this sense, the whole procedure is really optimized for large number of cells and moderate numbers of mutation. And we're using a Markov chain Monte Carlo scheme to sample the tree space. I will not go into this, but it uses sort of standard ideas from phylogenetic trees, or from Bayesian inference of phylogenetic trees. Let me show you one initial example. This was a very early single-cell sequencing data set published 2014, only 47 cells, 40 mutations, and 1.4% missing data. The authors of that paper estimated the false positive and false negative rate by control experiments, and they found something on the order of 10 to the minus 6 for the false positives and something close to 10% for the false negatives. And that's again a very typical thing for these single-cell DNA sequencing data. You have large false negatives rate, you're missing a lot of mutations, and false positives are typically not nearly as bad. So we're sampling from the posterior distribution over all trees, but I'm showing you only one optimal tree here because that's kind of much easier to do. And in this representation, I'm displaying the mutation tree. I'm also displaying the cells, these gray dots here, and I'm showing you the maximum likelihood attachment of the cells. Again, for the inference, I did not use it, but at the end of the day, if you would like to know what is the most likely placement of the cell, you can do that, of course. And I'm also putting in these blue boxes all mutations whose order I cannot infer. So here, for example, I'm only predicting, well, these 8 or so mutations occurred first, and then these 2, and then the next 3, and so on. But I have, within each of these blue boxes, I cannot resolve the order. So most of the cells attach towards the bottom of the tree, and there's only very few cells in this initial part that has a very linear structure. So my interpretation is that this linear structure corresponds to past clonal expansions with very few cells that are sort of left over. These were probably sweeps through the population. And then what we're seeing towards the bottom of the tree is the more recent coexisting subclones. One other example from a more recent data set that actually we didn't compute ourselves, but the authors of this paper used our tool to do it. So here they did not only have data from the primary tumor, but also from 2 metastases. And it's kind of an interesting example. First of all, there's a large number of cells attached to the root of the tree that's labeled zygote here. So these are all normal cells, or at least cells that don't have any of the somatic mutations. And then there's these 2 independent lineages that they found, shown in these bluish colors. And the 2 metastases have branched off the primary tumor in light blue at different steps of this evolutionary process. So very nicely can you reconstruct at which point in this progression the different metastases have branched off and the particular genotypes found in the metastases. So I want to go back now to the problem of mutation calling. I started with this binary matrix, but the question is, of course, well, where does the binary matrix come from in the first place? After all, it's a sequencing experiment, so really what you're having is read data that comes from the sequencer. And in an ideal situation where you have these 4 cells that have different mutational patterns, it may look like this. For example, the top cell has this blue and this greenish mutation. And what you're expecting for a heterozygous mutation, normal copy number status, let's say, is that roughly 50% of the reads have that mutation, right? So in an ideal world, this is what it would look like, and you would know how to deal with it. And in fact, from there, you could directly set up your binary matrix and everything is fine. Now, the reality is very different. The coverage is very uneven. You will miss positions or have very few coverage at some positions. You will often have quite a bias in the allele frequency. It may be far away from 50% and so on. And on the other hand, you would like to make inference now about the phylogenetic tree based on this. So while it's in a way clear that you need good mutation calls in order to get to a good tree, the reverse is also true. So suppose you knew the tree in advance, you would be in a much better position to call the mutations. And the idea is then to do this inference jointly, to make inference about the mutation calls and the structure of the tree at the same time. And the hope is that you're getting both better trees, but primarily better mutation calls in this manner. So one major ingredient of the model now is, first of all, you have to model read counts explicitly, so you need some distribution of read counts, and we use a beta binomial distribution. You have to account for over-dispersion in some way. And the other thing is that you need some model about the amplification process. David mentioned already that most of the problems really come from the amplification process. And here's a simple model of this MDA, multiple displacement amplification. So first of all, and this is real data on the right-hand side, a histogram just of the variant allele frequencies. And remember, for a heterozygous mutation, probably most of the somatic mutations are heterozygous. In any case, there will be many. You're expecting a frequency of 1/2, right? 0.5. So in other words, again, in an ideal world, you would see here a peak at 0.5. And as you see, there is no such peak, right? There's almost nothing. It's almost flat. So what you're seeing is peaks close to 0 and close to 1. But your ideal peak at 1/2, basically does not exist. And the reason is that most of the time you're seeing something, you don't see the mutation at all, or you're seeing it clonal. And that's what you have to account for. And you can think of this process as a very simple urn model. So suppose you have 2 different alleles, the blue one and the red one here. Now MDA, roughly speaking, you're picking randomly and you try to amplify. So suppose you take the blue one, you copy it, you amplify, and next step you amplified it again, and then maybe you pick the red one and you amplify it, and maybe again the blue one. So that doesn't look too bad. The allele frequency here would be skewed, but definitely you would pick up both of the alleles. On the other hand, if you managed to amplify only one of the two alleles, so the other one has dropped out, allelic dropout, well then you amplify this guy, and the problem now is that everything looks good because you have data at this position, you have reads, and you have no indication whatsoever that anything went wrong. Everything looks perfect, but you miss the mutation entirely, and that gives rise to these high false negative rates. So you need to model this distribution. I will not go into the details, but it's a mixture distribution because you have to account for the homozygous mutations also, and then the heterozygous one gives you this little bump here in the middle. This is what is left over from the ideal case, accounting for this amplification process. And that's what we're using in the model. Now I will go a bit quicker over results, but we compared now the performance of this method only in terms of the mutation calls, not in terms of the quality of the tree. And we compared it to MONUVA, that's another single cell mutation calling method that hardly shares any data between cells. So let's say roughly it does not share any data between cells, whereas our method, the main idea is to share information across cells via the phylogenetic tree. The tree describes the dependency structure. And if you look in the middle, that's precision, and the precision is very high for both of these methods, so they're very conservative, both of them. But in terms of recall, on the left here, you're seeing a huge gain due to the use of the tree and the simultaneous inference of the two. And we analyzed this as a function of the dropout rate. Of course, then the performance decreases, still much better for sci-fi as compared to MONUVA. Let's skip the one in the middle. The one on the bottom right is maybe interesting. We are not accounting for deviations of the expected allele frequency due to copy number changes, which in reality will exist. And so we're just assessing here how robust it is. And of course there is a drop in performance, but overall it looks pretty good even in the presence of copy number changes. So just to see what this looks like, so here's one example where the color coding refers to the posterior probability of a mutation call. So blue means we're almost certain it's a mutation. White means we're almost certain it's not a mutation. On the left is the mono-var data, just followed by hierarchical clustering to group a little bit the cells. In the middle is the mono-var calls, and then used only our tree building method, which is called SITE. And on the right-hand side is sci-fi, so the joint inference of mutation calls and tree. And as you see, the picture becomes basically clearer and clearer. Eventually you end up with a nice block structure that really gives you a very clear call for most of the mutations in most of the cells, and they nicely organize into the tree. So you're gaining a lot here simply by imposing the constraint of a tree. You don't know which tree it is, you have to infer it, but just the fact that there is a tree, assuming that there is a tree, gives you a lot of power in improving these mutation calls. Good. The last thing I want to talk about is also integrating bulk sequencing data into this inference process. So first of all, David mentioned that if you operate on bulk sequencing data alone, well, he said that's a very tough problem. I mean, I would say it's almost a hopeless problem. It's really, you know, in most cases, there's just not enough information in bulk sequencing data about the branching structure of a tree. Nevertheless, many of these sequencing studies, at least for now, are designed in such a way that people do a bulk sequencing experiment and single cells in addition. So if you have both, you can ask, well, can we maybe integrate the two and improve? So first of all, this is a data set from a panel sequencing approach from Steve Quake's lab, 2014 ALL, where they did this. And first of all, I'm just comparing here the tree we got using only the bulk data or only the single cell sequencing data. And you see it looks already quite different. I mean, they both have sort of two branches kind of, and they both have the same initial event on the top, but then these two branches look very different. The bulk tree tries to group things into clones, as we learned one has to do, and it has been done here, and the single cell tree is on the right-hand side. Looks quite different. So how can we combine the two? Well, I will not go into a lot of details here, but the point is now that you need to write down a joint likelihood for the two. So this joint score here, well, it's just a composition. I mean, think of this as log likelihood for the bulk data and the single cell data. For the single cell data, we use exactly the site model, as I explained it to you before. And for the bulk data, you have to, in one or another way, you have to go through this deconvolution exercise. And you somehow have to explain your observed allele frequencies as the sum over all clones harboring that particular allele weighted by the frequency of that allele. That's always sort of the main, like a matrix multiplication type of thing. That is the operation that describes the convolution, and you have to do inference on that, reverse, so deconvolution of it. And depending on how you formulate it, we assumed here a Gaussian distribution for the allele frequencies, and then the maximum likelihood problem for the bulk data becomes a quadratic program, such that we augmented now effectively our likelihood computation by this quadratic program. And that method we assessed now in terms of two criteria. The first one is the quality of the mutation clusters. So how good is this at inferring the clonal structure of the sample, compared here to ddclone. That's one of these methods for bulk sequencing data. OncoNem and B-site is our method, and I will not explain everything here. But these different lambda values on the top, the different boxes, refer to different amounts of noise that we add here to the allele frequencies. And you see that the combined method outperforms these two. And the second criterion is now again the accuracy of tree reconstruction. There's some measure for it, and again ordered by the allele frequency distortion. And again, you see that you have a benefit, especially if there's a lot of noise in the allele frequencies on the very left of the plot, small values of lambda, then you have a huge benefit by combining these two pieces of information. And so here's now the result, coming back to the earlier example, bulk on the left and single cells in the middle, as I showed you before, and on the right now is the combined inference for bulk and single cell sequencing data. And of course, this is only one example, but it's really a typical example in the sense that most of the information about the branching structure is really in the single cells. What the bulk data can be useful at is in estimating allele frequencies and in reordering linear stretches of the tree. If you're doing the bulk sequencing, especially with a reasonable deep coverage, then you can get better estimates of the allele frequencies than you can from the single cell sequencing data. And again, that's because of the amplification process. And so combining it, you can basically augment, I would almost think of it as augmenting the single cell inference by improved estimates of the allele frequencies. And that works, can work very nicely. Good, I want to wrap up here. So single cell sequencing data allows for reconstructing the evolutionary history and clonal composition of individual tumors if confounding factors are accounted for. So this is really the key. If you ignore all of these problems, then you are lost. Joint mutation calling and tree inference improves the accuracy of mutation calls in single cells. And the matched bulk and single cell DNA sequencing data can further improve both the quality of the trees and of the clones that you infer from the data. So all our software is open source and available. And I just want to thank Katharina Jahn, Jack Kuipers, and also Jochen Singer, who left my lab recently. They did most of the work, and the joint bulk and single cell inference was done together with Salem and Cenk. Thank you for listening.
Okay, any questions? I wanted to ask you, with the trees you built, is it possible to identify like driver mutations or positive selection events? Other than so you reconstruct the mutations that take place, the sequence of events, but those that may, be more important for cancer progression or that may be metastatic. Can you find ways to find them in your trees?
I mean, we didn't, okay, this is a matter of which mutations you put into the tree, of course. Right. And I mean, the truth is we didn't pay a lot of attention to this. The very first example I showed you, we just used the mutations that the authors, sorry for going through this, yeah, this one, we just used the mutations that the authors of the original paper identified, but these are mutations that they somehow deemed relevant for the process. These were 40 or so mutations in total, I think, in the beginning. And indeed, the coloring here is from us, so these yellow nodes, I didn't mention it, so those are known driver genes. And so it looks kind of nice in the sense that, you know, of course, this can, let's take the top 8 mutations there. It might be that these have been several clonal expansions, but it might also be that this has been one clonal expansion, maybe driven by this driver mutation, and the other ones are just hitchhiking, right? And they co-occur on this clone. So you can try to look at these things, but we didn't do it in any systematic way. And other than that, we usually just pre-filter mutations for being like completely uninformative for the phylogeny. Then, of course, we throw them out. But other than that, we just use the mutations that we find.
So how many mutations, out of curiosity, how many mutations are you considering normally for these trees? And
yeah, it's hard to say because it's a Markov chain Monte Carlo method. Right. So you can always start it. And you can always stop it, but whether it has reached convergence, that's the interesting question. And that in itself is difficult to assess, right? So you typically don't know. So we definitely did experiments with hundreds and thousands of cells, but the mutations are typically on the order of at most 100 or a few hundred, but more typically 50, 70 or something like this. I'm not saying these are the limits, but there are computational limits at some point, but the real question is how long do you have to run your Markov chain to reach convergence? And that's a tough question in itself, unfortunately.
Okay, any other questions?
In your model, you assume that every mutation has the same probability to occur, and thus can this influence somehow the order of the mutation that you infer?
You mean in the mutation calling model, the sci-fi, where we...
Yeah, you say you work with mutations in general, no? So I wonder if you were, for example, take into account that DC is more or less likely than AT or something like that.
Yeah, I see. It's a good point. No, we do not. I mean, let's say in the model where we try to call the mutations, right? There, it's again a full Bayesian approach. So we sample from the joint posterior of mutation calls and trees. And there, in principle, you could incorporate a prior about these mutations. But we don't. Or let's say it's flat, right? But that would be the right place to do this. If you have sort of an a priori guess and you say certain regions of the genome, for whatever reason, there's a higher chance to see a mutation, that would be the way to incorporate it. So it's a good point, but we're not doing it right now.
And you cannot guess these mutation rates from the data?
No, I mean, I can only, what I'm getting is the posterior, right? I'm getting afterwards the probability that there was a mutation or not. And of course, in principle, if I then have many of these samples, I can look across those and see what I found. But in the first place, you know, the prior has to come from somewhere else if you want to use one. Yeah.
Thank you.
Anyone else? Okay, you want to have coffee, right? Okay, thank you very much.